first slide you can see the anatomic location of the seminal vesicle, and the sections also contain, as you can see from this drawing line, the terminal portion, the oculary part of the ductus deferens. So the ductus deferens, you have seen it in the previous specimen, in the spermatocord specimen, and then now we reach its terminal part. It will unite with the seminal vesicle and continue into next tubular structure, the ejaculatory duct. So in this way, intersection, as the two organs approach each other, we will see both. In one of the specimens, these are a little bit further away, so easy to distinguish the two organs. And in the other version, they are very close, so, and also the structures resemble, so that won't be that easy. Now, here you can see what is the task of the seminal vesicle. So it produces, it's a secretory glandular organ. And the main products you can see here, the fructose, the ascorbic acid, prostaglandins, other sugars and amino acids. So these are the major products, and this will be added to the semen. Okay, so now let's see the sections. I already opened and show you where you can find these specimens. So the folder where you have to find these slides is week 10 and A seminar. So week 10 and A seminar is where you have to look for this specimen. 38 is the number of the seminal vesicle, and there is a second version, 68 per 1, and then for the prostate 96, and prostate, that is the other with the Gordner's prostate. Okay, so let's see the slides, going back to the panoramic viewer. So one specimen, seminal vesicle, but I would like to start with this one. This is where the two organs are not very close to each other, but we can see them in one specimen. So both of these structures are surrounded by a common adventitial layer. So they are embedded into a loose connective tissue layer. And then you can see as the upper structure, the oculary part of the ductus deferens. And then this lower portion shows the seminal vesicle. The seminal vesicle is not quite regular structure, so there are some invaginations of the connective tissue and other parts into the lumen. And in this case, due to the sectioning plane, there are seemingly separate luminated structures, but these all belong to the seminal vesicle. Okay, so these are the two structures. Now let's start with the oculary ductus deferens, and then you can observe what will be the changes between this segment and the previous segment, which is visible in the section of the spermatocord. Okay, so first of all, what we can easily recognize that the mucosal membrane is changed. Mucosal membrane was not a straight surface, but a more smooth surface than in this segment. Lamina propria shows folds, and the epithelium of the mucosa also covers it. So in this way, the mucosa and the lumen of the ampullary part of the ductus deferens has a kind of labyrinthic appearance. Okay, so then let's focus on it. Unfortunately, in some parts, the epithelium is disrupted. I think that is due to the section preparation procedure, so it's an artifact that epithelial layers are detaching from the surface. So the folds have a fold of the lamina propria and epithelial lining. We can see here what type of epithelium. The epithelium is same type as in the other segment. This is the pseudostratified columnar epithelium. And there are also ciliae here, though we cannot see their entire extent. I think that is also an artifact. What kind of cilia are present here? Stereociliae. Okay, so the epithelium mucosae is a pseudostratified columnar epithelium with stereociliae. Laminotropria is the next layer, connective tissue. Many blood vessels you can also recognize in the connective tissue layer, and it has some folds and side branches, so it has a kind of irregular appearance in this way. And then we reach the end of the laminotropia, approximately here. And now if you look at the muscle layer, though it's not so regular, but maybe in this part you can see the muscularis externa layer. What do you think, how many layers are visible here? There is only 2 layers. So instead of the 3 layers in the previous segment, in the spermatic cord, we see now just 2 sublayers of the muscle. What is the orientation of this inner? No, in this case it's circular for the inner layer and longitudinal for the outer layer. And inner longitudinal layer is missing. That is gradually disappearing in this segment. So muscle has 2 sublayers, inner circular and outer longitudinal sublayers. And beyond that we find the adventitia, the loose connective tissue layer. Okay, so this was the ductus deferens. Now let's see the seminal vesicle. The mucosa also labyrinthic, so the connective tissue layer shows the folds, same as in the ductus deferens, but you can see, if I go back into this view field, that the muscle layer is obviously thinner in case of the seminal vesicle. Layers are not so regular, but officially it's also an inner circular and outer longitudinal sublayer. Okay, but now let's see the mucosa. The epithelial lining of the seminal vesicle is a secretory epithelium. And these cells produce all these products, what I already mentioned, like the fructose, the prostaglandins, and so on. And the secretory mechanism is an apocrine-like secretory mechanism. And in this way, if you look at the epithelial lining, you can see some shorter cuboidal cells among the lining epithelial cells, and then some taller cells, and even some pseudostratified portions of the epithelium you can see. Here you can see nicely the columnar cells, and then it is changing to cuboidal cells at the base of this fold, and then again columnar cells. Okay, so this is secretory epithelium. And then the muscle layer, as I told you, it's not so regular, but it has an inner circular and outer longitudinal sublayer. Sometimes you can see some interwoven smooth muscle components, and that can disrupt the regular layers of the smooth muscle. Okay, so this is the seminal vesicle. Then in the adventitia layer, you can see some structure. What do you think about this one? That is a ganglion. A ganglion where you can see ganglion cells surrounded by what are these small cells surrounding these are the satellite cells. Yes?
Sorry. You mentioned the apocrine function, right?
Yes, so the secretory mechanism, this is a gland, the seminal vesicle, so the major function is the production of those material what I mentioned. And the secretory mechanism based on the morphology of the cells, the variable size of the cells is a histocrine-like secretory mechanism.
Not exocrine outside, like
Exocrine is general, yes, and then among the exocrine glands we have the three versions, the merocrine, the apocrine, and the holocrine, and this one is apocrine-like mechanism, what we see. So some parts, at least for the secretory material, accumulate in the apical portion of the cells, and then it is released together with some part of the cytoplasm. That is the definition of the apocrine secretion. Okay, so this is the basis for here. Okay, so then let's see the other version of this specimen. This is the one, and this is a segment which shows when the seminal vesicle is close to the ductus deferens. Yes?
You talked about the ganglia and the mucosa. You talked about...
Yes, I showed you the ganglions, yes?
Yeah, but then you started to talk about the cells around.
Yes, because there was a question here about the secretory mechanism.
So you can go back and talk about it again?
Yes, this is what I wanted to tell about the ganglion. Okay, so then we can assume that this smaller upper structure, that is the ductus deferens, and the rest of the luminated structures belong to the seminal vesicle. So you can see that segment where these two organs are very close to each other, and they will eventually unite to form the ejaculatory duct. Okay. Those students who are not interested in the class, you can leave, and there will be no consequence. If you would rather be in the full day, then please go, but don't disturb my class with talking in the background. It disturbs me. Many of my colleagues don't mind, but I mind. So either you remain silent or leave the classroom. Okay, so similar image, you can see here the mucosa shows folds, core given by the lamina propria layer and covered by the pseudostratified columnar epithelium. You can see some yellowish substance here that is most probably for lipofuscin pigment, which can accumulate in some of the cells. Okay, so that is the lamina propria lipofuscin pigment. Lipofuscin. You already heard of this. This is also called as the aging pigment. It accumulates in cells which have a long lifespan, like the, do you remember last semester we have seen it in the cardiac muscle, cells. Okay, so that is the mucosa, and then the muscle layer again shows the inner circle of the longitudinal sublayer, so it's the same as in the previous specimen. And when I enter the other part of the specimen, we can see the seminal vesicle. In the lumen, you can see some classifications. Most probably it includes the product of the seminal vesicle, as amino acids and some proteins are in it. Then it has an eosinophilic coloration. If you see cells, that is most probably an artifact during the sectioning. Some of the epithelial cells are shed into the lumen of the seminal vesicle, and that is what you can see there. So then mucosal membrane, same appearance, so it has labyrinthine picture, and the lamina propria layer is covered by the variable type of the epithelial cells. Mostly in this specimen I can see cuboidal lining epithelial cells, but we may find some areas where the cells are taller and they show the columnar appearance. Maybe here these are some taller epithelial cells sitting on the basement membrane and lamina propria layer. And then beyond that, after the mucosa, we find the muscle layer, and that is again the two sublayers, what I already mentioned. Okay, so these are the two sections. And now I will continue with the prostate gland. That is not a very complicated specimen, yes?
Is there a reason why the staining in the second slide for the epithelial surface of the vesicle lighter than in the first slide?
This is maybe just because of the different specimen and different staining, little bit different staining. You know, these are empirical staining, so then sometimes the staining is shorter time, longer time, then it can influence the intensity of the staining. Yes?
Where the non-appropriate or the healing sort of like circular structures, these ones?
These are also epithelial cells, but because of this labyrinthine structure of the mucosa, due to the sectioning plane, we can see them as separate structures, but otherwise they open into the main lumen also. So it's just because of the sectioning plane that you see it like that. Okay, anything else? Maybe connection with this specimen? Yes?
In the handout, the description of the healing of the surface, it says that it should specify when not experiencing. Is that the case?
In the previous specimen, the stereocilia were obviously visible, their number decreasing. Maybe a few of them you can recognize. So the number of the stereocilia is decreasing, obviously. That is another change in the different segments. But some few you can still see on the epithelial cells. Sorry? Decreasing the density of the stereocilia, increasing as we get into the more distal portions of the ductus deferens. Okay, so then let's change to the prostate gland. So that is that will be the next slide, and before I show you this specimen, I show you a summary image. So prostate gland is penetrated by tubular structures. The urethra we mentioned earlier, I think the prostatic part of the urethra is going through the prostate gland. It will receive the secretions of the tubular glands of the prostate, and also the ejaculatory ducts, which I mentioned, also travel through the prostate and then eventually drain or open into the prostatic urethra. Different zones we can recognize in the prostate gland, and it's occupied by tubular glands. A major part of the glands is peripherally located in the peripheral zone. These are also called as the main glands of the prostate, and they take around 70% of the total glandular units of the prostate. The parenchyma of the prostate is made of these tubular secretory units. Again, these have an evocline-like secretory mechanism, similar as for the seminal vesicle, and it has also stroma, a fibromuscular stroma, which is a mixture of connective tissue and smooth muscles. So this is what we will see, and it has also a connective tissue capsule that is also visible in our specimen. I also put here that the cancers of prostate are among the most common cancers in males, and one of the products of the prostate is used as a clinical sign. Its elevated level is a clinical sign of a possible cancerous alterations in the specimen. In the prostate, it's a prostate-specific antigen. That can be measured from blood, so it's relatively easy to measure it and collect the sample. If the levels are increased, then further experiments are necessary to identify if there is really a cancerous situation in the prostate. Okay, so what other products are produced by the prostate? Fructose, obviously, which is necessary for the sperm cells as their energy source, and also amino acids, sugars, prostaglandins are among the products of the prostate gland. And also prostate, beyond the prostate-specific antigen, it also produces prostate acid phosphatase, an enzyme, and that can dilute the semen, so that can liquefy the semen by its enzymatic activity. Also fibrinolysis, citric acid, these are the components of the products. Yes? The seminal fluid is initially produced by the testis itself. So it produces the sperm cells, and the fluid is also produced by the cell cells. Then it is altered in the epididymis. The duct of epididymis will reabsorb some parts of this fluid. Then additional secretions are coming from the seminal vesicle and the prostate gland. So these are the major sources of the semen, the seminal or the seminal fluid. Right. Now let's see the specimen itself. We have two versions, as I told you, one with hematoxylin staining. If you look at the specimen, you can see E-cut surfaces on the upper surface and the side surfaces, and you can also see the connective tissue capsule of the prostate on the lower surface of the specimen. We do not see the urethra itself, so the specimen does not include the urethra, so we cannot exactly tell the layers, but as we see the connective tissue capsule, we can say that most probably these glands, what you see here in high density, these belong to the main glands of the prostate. Okay, so these are the parts. Main glands of the prostate are the sites where typically cancers of the prostate appear. So this is the peripheral layer. Now let's see more of the details. Connective tissue capsule, it's mostly formed of collagen fibers, connective tissue cells, so fibrocytes are there. Then peripheral fiber bundles and small-sized blood vessels you can also recognize. And then we enter the prostate itself, and in between the secretory units, you can see this mixed tissue, the fibromuscular stroma of the prostate. I would like to find an area where we can see what are the components of this tissue. So you can see the collagen fibers here as part of the connective tissue components, and you can see cells with very dense staining and flat cell nuclei. What do you think? What kind of cells are these? Fibrocytes. Yes, so this is one typical cell, and if I come to this region, then you can see cells with elongated nucleus with euchromatic staining. What do you think? What kind of cells are these? These are the smooth muscle cells. So in this way, we can see both cell types, smooth muscle cells and fibrocytes. and also the extracellular matrix of the connective tissue like collagen fibers. You can also see some unstained areas that is most probably occupied by the ground substance molecules. Okay, so fibromuscular stroma, this is what we can see. And then embedded into this stroma, we can see the tubular glands. These tubular, some of them contain certain structures, but let's find first the gland where we do not see any content in the lumen. So these are also secretory cells, glandular epithelial cells lining the lumen. And you can see a similar phenomenon or similar morphology here as in the seminal vesicle, that the shape, the size of the epithelial cells is changing. So there are some shorter cells, even sometimes in some areas of the lining epithelium you can see low cuboidal cells, like for example here, and then gradually taller cells, cuboidal, columnar cells, and sometimes pseudostratified epithelial areas you can also recognize with basal cells and tall secretory cells. So the secretion is again apocrine-like, as we defined in connection with the seminal vesicle. The secretory product typically accumulates in the apical portion of the cells and then released into the lumen together with some portion of the cytoplasm as well. So it's an apocrine-like secretion, what we can see here, and the morphological basis of this kind of secretion. Okay, so these are the regular glands, tubular glands. And in some of these glands, you can identify these rounded or sometimes oval-shaped structures. These are concretions produced by the precipitation of the secretions of the prostate, also called as corpora amylacea. And these, the larger ones especially, they can calcify also. So when you see in the central part of these structures the basophilic staining, that is a sign of the calcification. Okay, so these are corpora amylacea. In the early childhood, these are not present in the prostate gland. They appear during the aging process, and their number is increasing. We can identify these structures in all gland types of the prostate gland. When they accumulate in high number, especially in the mucosal glands of the prostate, they may cause enlargement of the prostate. That is quite common among males after the ages of 60, and because of this enlargement of the prostate tissue, then the urethra can be compressed, so this can cause problems in urination. But that is an age-related change. Also in the main glands, you can identify these structures in the specimen. Many of the corpora amylacea you can see. Okay, so I come here back, and let's see the last specimen. That is again prostate gland, but with the golden trichrome staining. I don't know if you remember, we already had specimens stained by this staining. Trichrome means that there has to be at least 3 colors for the staining. If you look at this specimen here, you can see the oriented differently than in the other specimen. The connective tissue capsule is visible here. I come to here and let's identify the colors provided by the Goldner staining. So greenish bluish color. What do you think? What structures are stained in this color? Collagen. It's mostly the collagen what is stained. If you remember the small intestine specimen, also the mucus in the goblet cells was stained in similar color. Okay, so one color. Then in the lumen of the blood vessels, we can see red blood cells, and now we can see this orange color. So orange is for what's the part of the specimen, that is for the cytoplasm in general, red blood cells here, yes, and then for most of the cells, the cytoplasmic staining is reddish orangey, so sometimes purple. That is the second color. And the third color is the staining of the nuclei in gray color. Okay, so these are the three colors. Sometimes it's not that obvious, but these are basically the staining effects of the coordinate trichrome staining. Okay, the reason why this specimen was included into the series of the sections, that with the aid of the coordinate staining, it is easier to distinguish between the muscular components of the stroma and the connective tissue parts. So if you look at this portion of the specimen, you can see the smooth muscle cells in at least partly in orange color, the nucleus is green, and you can easier distinguish between the smooth muscle and the collagen fibers with the aid of this staining technique. Okay, so the fibromuscular stroma is the main point in the usage of this kind of staining. So connective tissue parts and smooth muscle components are easier to distinguish in this case. Now let's see the glands. Again, we can see glands, tubular glands with irregular lumen due to the different shape and size of the lining epithelial cells. And you can see also the concretions in high number in the lumen of some of the secretory units. And the peripheral part contains mostly organic components, that is this bluish-greenish color, and the centrally calcified portion is changing its color to more reddish or at least darker coloration, like in this example of the concretion. Okay, so that is the next specimen, yes?
So this is one staining, that's Goldner.
That is called as the coordinate trichrome staining. This is one protocol for the staining. Of course, different chemicals are used for the staining technique.
So it's not just one,
No, it's not just one dye, but it's a combination of dyes. I don't know exactly what are the components, and I have never done this protocol. But it's different kind of dyes. When you go to pathology, histopathology, you will learn more trichrome stainings. So you will see more of these type of stainings when at least three structures are differently stained by different color. Okay.
Do we have any elastic fibers, or is it just collagen?
As far as I know, it's mostly collagen fibers. We cannot exclude that in low amount there can be some reticular fibers or maybe some elastic fibers, but obviously majority of the fibers are collagen fibers.
Is the space around the foot biological or it's not?
Around the corpora cell, it's maybe natural because many of these structures, they don't fill the entire lumen. So they start as small precipitations. Maybe shell epithelial cells can be the starting point of the precipitation of the secretions, and then gradually more and more layers are added to these structures. So they not necessarily fill the entire lumen. Then I give you now some time to look at the specimen and ask your questions, and then pour specimen to the screen.
Yeah, Like if you just compare them together, what are like the major differences?
Basically, these are not that different, but if you look at the two of them together in small, then you will see that in the upper case, the two organs are in a distance, so there is obviously a layer of adventitia in between, loose connective tissue in between the two organs. While the lower one shows when they are very close to each other, so there is just a very, very narrow layer in between the two structures. Otherwise, these are basically the same.
And you said the first section is closer to the axillary region, but what about the second section?
The upper one, that is where the two structures are still within a distance. And the lower one is when they approach each other. They are very close to each other. Yes? Yes, yes. So if you look at trying to catch a frontal view field where good examples for the two cells we can see. So basically the nucleus is what we can see, and they differ obviously. We sometimes see the entire extent of the smooth muscle cells, but sometimes not. If you look at these cells, then on the right hand side you can see two cells with the euchromatic nuclei. These are the smooth muscle cells. And on the left hand side, these two have heterochromatic nuclei. These are fibrocytes. Okay, so in this way you can distinguish the two cell types. Okay. Alright, so then a few minutes to look at this lesson. Let's find then renal corpuscle. This one is where you can see the two poles of the renal corpuscle. That is the first part of the nephron. The vascular pore you can see here. The vas afferent is visible, vas efferent is not so obvious. The glomerulus, and then it is surrounded by, do you remember what the Bowman's capsule, and the which layer is by the podocytes? Do you remember that is the visceral layer, is the podocytes, and the parietal layer is the simple squamous epithelium. Okay, so this is the renal corpuscle, yes? The podocytes you can find on the surface of the capillaries. This nucleus, for example, I think it's quite obviously a nucleus belonging to a podocyte, because that is larger than the surrounding cells nuclei, and it is euchromatic. So podocyte, we don't see the processes which are nicely visible on electron microscopic images, but we can see the nucleus. Also here you can see these light staining cell nuclei. Endothelial cells are obviously different. They have the heterochromatic staining and very flat shape, so these are endothelial cells. And there is a third cell type among the loops of the capillary. Do you remember what are those cells which can serve as supporting cells here, but for the mesangial cells? The mesangial cells, their nucleus is that is the least obvious. If you find some small oval or round-shaped cell nuclei among the capillary loops, these may belong to the mesangial cells. Okay, so mesangial cells the endothelial cells lining the capillary loops and the podocytes you can see on the surface of the capillaries. These are the three cell types in this area. Okay, then this is the vascular pool where the vas afferent comes in, and this is the urinary pool where the tubular system starts. This is the proximal convoluted tubule, so that is the first segment. Proximal tubules, epithelial cells are cuboidal and they have a strong eosinophilic staining. Also they bear microvilli on their apical surface, and that will make the lumen of these proximal tubules irregular. Okay, so proximal convoluted tubule first. Then the proximal convoluted tubule continues into a straight segment, and the straight segments we find here. What do you remember? What is the name of these? These are the medullary rays. Yes. So in medullary rays we can find the straight segment of the proximal tubules. That is the next part of the nephron. And then these form the descending limb of the loop of Henle. So they will travel towards the medulla. You still see medullary ray. Now we enter the medulla, and there are gradual changes in the lining epithelium. So it becomes shorter. This is still showing the typical morphology of the proximal tubule. These are all straight segments, what we find in the medullary part. And then it continues into the thin segment of the loop of Henle. I go through the most of the medulla and travel to the papillary part. And here you can see the thin segment, which is lined by a simple squamous epithelium. So there is a gradual change from the cuboidal epithelium to the simple squamous epithelium in the thin segment of the loop of Henle. That will make a hairpin turn and move back towards the cortical region. So thin segment in the medullary part. And then it returns towards the cortex. So as I travel further, we can identify the straight segment of the distal tubule now. And the straight segment of the distal tubule we can recognize based on cuboidal lining, epithelial cells, a lighter staining of the cytoplasm, and a more regular lumen. But this will be more obvious when I enter the medullary ray, where we can see these tubules. So left-hand side one is a proximal tubule, and this one is a distal straight tubule. This is part of the ascending limb of the loop of Henle. Okay, so distal straight tubule. And then the distal tubule leaves the medullary ray, enters the cortical labyrinth, and the convoluted segment will be the next. So among the renal corpuscles and the proximal tubules, we can identify the distal tubules, the convoluted segment of these distal tubules you can see. And if you remember, the distal tubules return to their nephrons, renal corpus hole, and at the vascular pole they form a special structure, something like this, what you can see here. What is that structure? Do you remember? Well, this is a spot-like structure.
Just what's near the beginning?
Yes, so the entire structure is called as the juxtaglomerular apparatus, And the structure, which is formed here by at one part of the juxtaglomerular apparatus, that is the macula densa. So that is a special region in the distal convoluted tubule where the cells are closer to each other, so there is a higher cell density, and this is the name of this macula densa again, and is part of the juxtaglomerular apparatus. Do you remember what is the function of these cells of the macula densa? These are receptors, chemoreceptors of sodium concentration in the urine, and they will signal to other components of the juxtaglomerular apparatus. And then what will be these other components? There is a secretory cell type in the juxtaglomerular apparatus that is modified smooth muscle cells, also called as juxtaglomerular cells. What is their product? Do you remember? Renin. Renin is the product. And there are also extraglomerular mesangial cells as part of this complex structure. So the receptor is the macula densa cells. The effector by the renin production is the modified smooth muscle cells, the juxtaglomerular cells, and what provides communication between the two is the extraglomerular mesangial cells. Okay, so convoluted distal tubule segment has this special spot-like structure, the macula densa. Then through a short, narrow tubule, the urine is conveyed into the collecting tubules, but the collecting tubules are not part of the nephron. But collecting tubules we can find in the medullary rays, typically in peripheral location, with larger lumen, taller cuboidal cells as lining, and the cell borders are mostly visible between these cells. So these are the morphological differences here. Yes?
I would like to know, is it true that it goes from low cuboidal to low columnar?
Yes, so in the cortical segment, they have single cuboidal epithelial lining, but as we travel into the medulla, to their terminal portion in the papilla of the pyramid, then they turn into columnar cells. So there is a very gradual change of the lining epithelium, and finally the terminal portions will be like the columnar epithelium.
Yeah, and can you talk a bit about the dark and light cells in the production duct? Aren't those physical modifications?
No, I cannot show you these, so they are not visible with the HND staining. I cannot tell you about that. Yes? Arteries. We can go through the blood vessel system as well. Largest vessels you can find here in the renal column. This one, if you look at it, it's not exactly in cross section in plane, but slightly obliquely section. But still, it's visible that it shows characteristics of an artery. So internal elastic membrane you see in the tunica media, mostly smooth muscle cells, and the border between tunica media and adventitia is obvious. So because of its location, we can say that this is the interlobar artery. Interlobar artery, that is the largest. Then the interlobar vessels, actually a vein is traveling parallel with the artery, but it's not visible in our section. Then they will make a turn at the border of the cortex and the medulla, and the next segment of these vessels is the arcuate vessels, if you remember. Again, we find an artery and a vein next to each other, so these are the arcuate artery and the arcuate vein here. Then For the supply of the cortex, we find as next segment interlobular arteries. I cannot show you so obviously these arteries because not in the sectioning plane, but an interlobular vein is visible here in the section. And time to time you can see also the artery which runs more or less parallel with it. So interlobular artery will be the next. Then from the interlobular artery, the next segment is the smaller arterioles, which will serve as the afferent arterioles of the renal corpuscles, so they will enter at the vascular pool. Then glomerulus, capillary bundle, and then the vessel which is leaving is again an arteriole. So that is an interesting circulatory system. It is also called as arterial porter system. And then the next part of the circulatory system depends on the location of the renal corpuscle. From these, where I am now, so for the superficial intermediate renal corpuscles, the was efferent will supply second capillary bed in between the tubules. So here, if you look at the in between the tubules, you can see some endothelial cells and blood vessels. Many times the lumen is empty, but sometimes you can see the formed elements of the blood in these capillaries. So there is a rich network here, peritubular capillary system that will receive the reabsorbed material from the tubules, and they are lined by fenestrated endothelium. Then what happens in case of those renal corpuscles which are close to the medulla? These are called as the juxtamedullary corpuscles or glomeruli, where the vas efferent is traveling. The vas efferent is supplying the medulla from these. So the juxtamedullary renal corpuscles vas efferent is not forming capillaries in the cortex, but it will go into the medulla and forms the straight vessels or vasa recta of the medulla. There will be an arterial segment in the superficial part of the medulla. In the deeper part, there are capillaries, and these capillaries are important in the formation of the final composition of the urine. There is an exchange system here between the lumpa fendae and the capillaries. This is also called as a counter current amplifier system. You will learn the physiology of this next year in the physiology subject. So the capillaries and the thin segments of the lumpa fendae we can also see. And then the venous part will ascend back, also will be part of the vasa recta, because these are also straight vessels. And there are two possibilities: either these veins directly drain into the arcuate vein, or some of them enter the cortex and drain into the interlobular veins. So these are the vessels. Okay, so was that your question, the vascular system? Okay, anything else? And then, okay, so then last part of the seminar, I wrote some PPTs to talk a bit about the development of the genital system. It has two parts, which is coming together at the point of the development. So gametogenesis, which you already discussed on the earlier seminars in connection with the ovary and the testis, and the development of the gonads. Okay, Do you know where the germ cells are coming from? You probably mentioned that earlier. The epiblast, and then they move as a second station to the yolk sac, and then they will travel towards the primordium of the gonad, and this next image shows that. This shows that step of the primordial germ cells when they are in the wall of the yolk sac. And then on this image you can see that through the serous membranes of the hind gut, they will travel to the primordium of the gonads. This is the so-called genital ridge that is appearing at the this is image I wanted to show to you, where it shows what is the location of this genital ridge. If we look at this next image, it shows when the germ cells, primordial germ cells, arrive to the gonad primordium through the mesentery, and they enter the indifferent gonad. The gonad primordium has basically two components. This is a fold of the lining epithelium on the posterior side of the abdominal cavity. Surface is covered by epithelium. This is also called as coelomic epithelium, and its core is given by mesenchymal tissue, condensed mesenchymal tissue. Epithelium is black undergoing, orange color is the mesenchymal tissue, and the red is the primordial germ cells. Okay, so these are visible. The epithelial lining is proliferating and forming kind of pores inside the primitive gonad. From now on, development is together for the gonad and the germ cells, and the germ cells influence how the further development is going on. It depends on the chromosomes which is carried by the germ cells. This is just the same. This is what I wanted to show you next. In males, there is a transcription factor called SRY, which will determine the development of the testis. This is missing in females, and whenever this gene and its product is missing, then ovaries will develop. So in the case when SRY is not present, then the indifferent gonad will develop into ovary. Okay, so this next table shows what happens. So the testis develop, and the cords from the epithelial cells develop in the cortex and medulla, and then later the cortical cords disappear, and the tunica albuginea also develops. Then, in case of ovary, the medullary cord degenerate. As you remember, the medullary part of the ovary does not contain oocytes and follicles. So, in the medulla part, we do not find these structures, and cortical cords develop to form the follicular epithelium, and no tunica albuginea in this case. Now, this next slide shows the development of testis, and again this is partly revision. Mesonephric duct you can see here as part of the mesonephros. So this is the mesonephric duct and the mesonephric tubule that is coming from the mesonephros, as I mentioned. And another tubular structure is also developing partly parallel with the mesonephric duct from the folding of the epithelial lining of the primordium of the gonad. This is called as the paramesonephric duct or Mullerian duct that will degenerate in males and that will form organs of the female genital tract in females. Now, if you look at this image, you can see that in case of the testis, the medullary cords develop and they contain the germ cells also. The end of these cords will form the retetestis, then they do not contain the primordial germ cells. Okay? On the right hand side picture you can see a later developmental stage. Here these cords form loops. Thus these original medullary cords, they will form the seminiferous tubules, the convoluted and the straight segment of these, and also the retetestis. Now I put here two cell types. What do you think where the Sertoli cells develop from? The possibilities are the cords, testis cords, or the mesenchyme in between them. So Sertoli cells come from the testis cords, from the epithelial cells, and lytic cells develop from the surrounding mesenchyme, the testosterone producing cells. Okay? And then if you look at the mesonephric duct, you can see that the excretory part will develop into the efferent tubules of the testis. So they will be part of the tubular system here, and also the ductus epididymis and the ductus deferens will develop from the mesonephric duct. Okay, one more slide. From an earlier chapter of your textbook, which shows the difference of the seminiferous tubules in a newborn on the left hand side and after puberty on the right hand side. You can see that before puberty, the lining of the seminiferous tubules is the primordial germ cells and the Sertoli cells, and the other cell types, the spermatocytes, spermatids, sperm cells are not present until puberty. Okay, so this is the male part, and now let's see female situation. So again, primordial germ cells arrive to the primordium of the ovary, and these primordial germ cells will turn into oogonia. By mitotic divisions, their number is also increasing. I put here these numbers. So in the 5th month, the number of the cells of the oogonia is around 7 million. And then after 5th month, the oogonia start to enter the first meiotic division and become primary oocytes. And these will stop in the trophies of the first meiotic division. But I think you already discussed that in connection with the histology of the ovary. And then many of the oocytes become apathetic, and then during the active period of a female, around 500 of these oocytes will be ovulated. Okay? And then how the gonad is developing after the arrival of the primordial germ cells. You can see that the cortical cords remain and the medullary cords degenerate, and then these cortical cords, the epithelium of the cortical cords, will develop into the follicular cells of the primordial follicle. So these will be flat epithelial cells until puberty again. So the primary oocyte will be surrounded by the squamous follicular cells, and these will develop only after puberty, after the influence of the FSH further. Also, factor oocyte maturation inhibitor acts and suppresses the further development of the follicles and oocytes. Then in females, the mesonephric duct mostly degenerates, except with some remnants. If you remember, we saw some remnants of the mesonephric duct in the specimen of the uterine tube. That is the echophoron or parophoron. These can be remnants in females. And then the Mullerian duct, that will develop further. What will be the derivatives of the paramesonephric duct? The fallopian tube, yes. And then by the unification of the Mullerian ducts from the two sides, the uterus is formed, so that will be a second derivative. And also the upper segment of the vagina is also developing from the Mullerian duct. Again, from a previous chapter, I wrote here this illustration showing how the arrangement of the oogonia and primary oocyte is changing during the development. So in the first months they form groups and they divide by mitotic divisions. In 7 months, these cells are mostly primary oocytes surrounded by the follicular cells, and then this is the newborn situation. And also on the surface we can still see the surface epithelium that is also visible on the specimen. If you remember, on the surface of the ovary we find cuboidal epithelium. Sometimes it is flattened, but officially it is a cuboidal epithelial lining. And this show the further development of these ducts. In the A is the male and B is the female situation, but these are not previously discussed. This figure shows the formation of the further derivatives of the paramesonephric duct. So the uterine tube, the uterus, and the upper segment of the vagina is a derivative of this tubular structure. In males, the paramesonephric duct degenerates. And also on this figure you can see those remnants which you also mentioned, the echophoron, the parophoron, and the Gärtner's cyst can be one more remaining vestigial structure next to the vagina. So these are the structures which develop here. The time is over for the seminar. So we will finish for today. Next week, as I know, as far as I know, you have the normal